When Charles Darwin published his book, The Origin of Species, the general concept was that even though there are variations like new breeds of dog or farmers were obviously breeding new strains of crop, that species more or less stayed fixed. There was an idea of, quote, the fixity of species. As species would not significantly change over time, and certainly no new species could arise. After Charles Darwin's publication, and then the works of Alfred Wallace and many other biologists, it was then appreciated the degree to which things change over time. Populations evolve. That old idea that the life that we see today, the pigeons, the goats, the gray squirrels, etc., they more or less existed in just the same way at the beginning of life on Earth, and then far into the future they would look about the same. We now know that this is not the case. Life is forever changing. But in biologists, we then study this. We make predictions. We test those. And so therefore there is a field of biology known as population genetics, because you can actually measure the genes which are affecting these anatomical traits, and then measure them to see if they are changing over time and see to what degree they're contributing to speciation, etc. So first statement, as I introduce an equation known as the Hardy-Weinberg equilibrium, is that evolution does not have to happen. Now, in this case, I'm going to just do a very simple example where we're dealing with, say, an incompletely dominant trait. So this individual is blue because they have two of these alleles. I know it says homozygous dominant. This will be an incompletely dominant trait. So this individual is blue because they have these two alleles. This person is yellow because they have these two alleles. Now, an allele is a form of a gene. You and I, we both have hair color genes. We both have, blood type genes. But just because we have the same gene doesn't mean the form of the gene is the same. I'm blood type O, you might be a different blood type. So even though we have the same gene, the alleles of the gene, my alleles make me blood type O, your alleles might make you a different blood type. And so these two individuals, they have an A gene, but they have different alleles. This person has two of these blue alleles. This person has two of these yellow alleles. Now, in this population, we can imagine that these two individuals represent the population as such. All of the individuals then are either yellow or blue. And if you were to count the number of alleles in the population, in this case here we see 4. This individual has 2 alleles, this individual has 2 alleles. And the blue allele is 2 of the 4, so half. And this allele is 2 of the 4, or half. Soon we'll be using terms P and Q where each is 0.5. Now, let's imagine it's an incompletely dominant trait, so that when they have offspring, if their offspring receive a blue allele from one parent and a yellow allele from another parent, that their outward appearance is green. So these children look completely different from their parents. They have a different phenotype. None of them are yellow, none of them are blue, they're all green. So the phenotype has changed, the outward appearance has changed. But let's look at the alleles. So there are 4 individuals. Each individual has 2 alleles. So there are 4 blue alleles in the population, 4 yellow alleles in the population. So the blue alleles are half of the total. All right, the yellow alleles are half of the total. So even though the phenotype is completely different from what it was in the previous generation, the allele frequencies are the same. All right, so all of these individuals then have one of each allele, and the blue allele and yellow allele are half of the alleles of the population each. Well, let's now look at what the next generation might produce. So if these two individuals have offspring, then a quarter of the offspring will receive two blue alleles. A quarter of the offspring will receive a blue from their father and yellow from their mother. A quarter of the offspring will receive a yellow from their father, a blue from their mother. A quarter of the offspring will receive a yellow from each parent. So if we look at the phenotypes, we would see a 1 to 2 to 1 ratio of blue to green to yellow. A quarter are blue, half are green, a quarter are yellow. But if you were to count the alleles in this population, so there are 4 individuals, 2 alleles each, so there are 8 in total. So this individual has 2 blues, 1 blue, 1 blue, so there are 4 blue alleles in the population. There are 4 yellow alleles in the population. So in each of these 3 generations, the phenotype has changed. We went from, half blue, half yellow to all green to half green, a quarter blue, a quarter yellow. Each of these generations has had a different phenotype ratio. But the genotype ratio, the allele frequency, has stayed the same. Now, this is important. We're not done with this scenario where evolution doesn't happen, because it didn't. If evolution is change over time, all right, and we had 50%, 50% allele frequency in the first generation and the second and the third, in that example, there was no change. Right? Even though the outward appearance changed, the alleles didn't, the population did not change. It still has the same genes and forms of the genes that it once did. There was no evolution in that population. So evolution does not have to occur. But as you can see there, to determine that, we're going to have to be able to measure the allele frequencies. So how would we do that? I'm going to introduce an equation, the Hardy-Weinberg equation, in which there are 2 alleles in a population, just as there was in the previous example. Let's call one P and one Q. And if there's a dominant and recessive, so let's deal with a dominant recessive example, not the incompletely dominant that I just saw. Let's say the P is the dominant allele and the Q is the recessive allele. So first I say P plus Q equals 1. In that, all of the alleles are either the P allele or the Q allele. All right. So when you add together all of the P alleles and all of the Q alleles, which in the previous example would be the blues and the yellows, that equals 1. That equals 100% of the alleles in the population. So the alleles of the population are just composed of these 2 plus. So P plus Q will equal the total. And as we'll see in just a second, we know from algebra that if we square an equation, if we square both sides, then we haven't changed the meaning of the equation. So if the equation is true, so if x equals y, once you square it, then x squared equals y squared. So you haven't changed it. And so if we square that equation, P plus Q equals 1, then we get P squared plus 2PQ plus Q squared equals 1. We'll see that in a second. So if P plus Q equals 1, then the square of that is true. We haven't changed the meaning. All right. So let's apply this. Let's say you're looking at a population of dogs. There are dogs with the dominant trait of having a darker coat and the recessive trait of having a yellow coat. And you would like to know what P and Q are because you would like to measure allele frequencies and potentially a change in allele frequencies over time. So let's say that we note that 3/4, 75% of the dogs have the dark coat, 25% of the dogs have the light coat. Great. Well, I'd love to say I'm measuring P plus Q directly, but remember that each individual can have 2 alleles, all right? And so say that dark dog, was it dark because it had 2 dark alleles? It would be dark, but if it had a dark allele and a light allele, if the dark allele is dominant, then we wouldn't know. So we're going to focus on the light colored dog. The only way to have a light color is if this dog got a light gene from its mother and its father. What are the odds of that? Well, the odds of that would be Q, all right, as we'll see. If the light allele has a certain frequency in the population, let's call that frequency Q, the odds that the father's sperm had that allele in it is Q, all right, its frequency in the population. The odds that the mother's ovum had that allele in it is Q, the frequency in the population. So the odds of getting this combination is Q squared. So Q squared is then the frequency of the light-colored dogs. Now this, see, this will make sense in just a second, but this means that when you study a population, you don't have to necessarily count these as much for now. We need to know that the light allele is 25% of the population. Because if that's Q squared, and you want to know what Q is, well, if Q squared is 0.25, then we can simply take the square root of this. The square root of 0.25 is 0.5. So we'd like to know what Q is. If you know that 25% of the population received a Q allele from their mother and their father, that's essentially Q squared. And so if Q squared is 0.25, 25%, then Q is 0.5. And so then we go back to that Hardy-Weinberg equilibrium. If P plus Q equals 1, all right, so if all of the alleles in the population, 100%, is equal to P plus Q, once we've determined that Q is 0.5, and you want to say, all right, well, what's the frequency of P in the population? What is the frequency of having a dark allele? Well, if 0.5 is Q and P plus 0.5 equals 1, then 1 minus Q is then equal to P. So P equals 0.5, okay? And so in this population, what we've just said is that P is 0.5. So the percentage of sperm having a P allele is 50%. The percentage of ovum having the P allele is 50%. If the frequency of Q is 0.5, the frequency of sperm having the Q allele is 50%, the frequency of the ova having the Q allele is 50%. And so then the odds that this dog will get a P allele from each parent is 0.5, which is the frequency of its being in the sperm, times 0.5, which is 25% of the dogs get a P allele from each parent. 25% get a P allele from their father, a Q allele from their mother, 25% get a Q from their mother, a P from their father, and 25% have a lighter color because they resulted from a sperm with the Q allele, that was 50% of the sperm, and an ovum with the Q allele, that was 50% of the ovum joining. And so this is how we get the Q squared value. The Q squared is those offspring resulting from the fusion of a sperm with the Q allele and an ovum with the Q allele. So if this is Q, what's the frequency of sperm having the Q allele? Q. What's the frequency of ova having the Q allele? Q. And so Q times Q is Q squared. So Q squared equals 25%. So P plus Q equals 1. All right, so the number of P alleles in the population plus the number of Q alleles in the population equals all of the alleles in the population. And if we then square this, we get P squared plus Q, 2PQ plus Q squared equals 1. And see, now the beauty of this is I can ask the question now, are all populations the same? Is this population changing over time? If I then measure another population and I say, ooh, this is different, all right? Here I'm getting 16% of the dogs have a light color, 84% have a dark color. Well, once again, I'm going to focus on the light color. If 16% have a light color code, I want to know what Q is. But the light colored dogs essentially are Q squared. All right, the only way you get a light colored dog is if sperm with the Q allele fused with ova with the Q allele. So it's Q times Q, Q squared. And so then I say, all right, well, if Q squared equals 16% of the population, 0.16, then what is Q equal to? And then you say, oh, well, let's just take the square root of that. The square root of 0.16 is 0.4. So 40% of the alleles in this population are the Q alleles. All right, so Q equals 0.4. What is P? Well, that's easy, because if P plus Q equals 1 and Q equals 0.4, then we can simply say 1 minus 0.4 equals what? Equals 0.6, which is then the frequency of the P allele. And so now, if 60% of the sperm have the P allele, 60% of the ova have the P allele, 40% of the sperm have Q, 40% of the ova have Q. This then gives us the equation which we noticed, where 16% of the population would then have a Q that they've inherited from each parent, and would then be Q squared. All right, and so now we've used this to calculate the values of P and Q in the population. It's 0.6 and 0.4. And once again, we use the square of this, P squared plus 2PQ plus Q squared equals 1. That's just the square of our original equation. Now, I go through more examples of this, and when I teach this in class, I would say no one understands what I'm talking about the first time through. But then by the next couple, they're like, oh, okay, this is easy. I get that. So all you need to do is say, all right, right now I'm going to look at those with the recessive trait, all right, because that's Q squared. What's the frequency? And so if now the individuals with the recessive trait are 9% of the population, that's Q squared. If Q squared equals 0.09, then we take the square root of both sides, and then you see that, oh, Q is 0.3. Once I know what Q is, it's easy to calculate the frequency of the P allele, the dark allele, because P plus Q equals 1. So if Q is 0.3, then P would be 1 minus 0.3, which is 0.7. All right, and then once again, we see this here. That's why 9% of the population has light color. So if 30% of the ova have the light colored Q allele, if 30% of the sperm, then this dog has a 0.3 chance of getting an ova with the Q allele, a 0.3 chance of getting a sperm with the Q allele. 0.3 times 0.3 is 0.09. This now depicts the alleles of our population. I'm going to jump forward, but I could do the same with other allele combinations. Obviously, I've been doing the easy ones that you could perform the square root in your head, but you could have instead of Q is 0.2 or 0.3, it could be 0.25. You would just have to use a calculator to do the square roots. So in population genetics now, we can measure allele frequencies, because that then gets us back to our original question. Is the allele frequency changing over time? You could make a prediction. Yes, it will. No, it won't. It is in this case, etc. But if you can measure it, now here's a way of mathematically testing this. This is a science, right? You can make predictions. You can test them. And getting back to what I just said, evolution does not have to occur. You can have populations whose allele frequencies are not changing over time. You can measure that. Once again, this is a science, and we can just state observable facts. The allele frequency is not changing over time, and by using the Hardy-Weinberg equilibrium, we can demonstrate that. But, here's the big but, the Hardy-Weinberg equilibrium, to always remain the same, needs certain conditions. It turns out you can measure these. Once again, this is a mathematical thing you can apply to populations. So you will not see evolution. You will not see a change over time if the population is infinitely large. I'll go through all of these. That if you have no net immigration or emigration with neighboring populations, if forget which one I do next, sorry. If mating is random, so if the females with one allele do not have a preference for males of a specific allele. If there is no natural selection, if it makes no difference whether you have the P or Q allele and how long you live, how well you can evade predators, your resistance to disease, there is no net mutation, so that the P allele is not mutating at a different rate from the Q allele. If P can sometimes mutate into Q, then the rate of Q mutating back into P is the same. So if you take a population with P and Q, and that these conditions are true of P and Q, and the population in general, then you would predict the population does not change over time. Evolution does not occur. You come back thousands of years later, and you will have the same allele frequencies in the population. However, these conditions are never met. Now, for example, with size, some populations are really big and some are tiny, so sometimes they come closer to being met, but they are never met. As a result, evolution has to occur mathematically, because the conditions that you would need for allele frequencies to not change are never met. It is mathematically impossible for you to come back thousands of years hence and find this dog population with the exact same alleles in the exact same frequencies as we currently have, because the conditions for having the alleles remain unchanged, they are not met. So then very quickly, then population geneticists, if they see that there is a change over time, they could ask why. Why is the frequency of P and Q changing over time? And one thing could be that mating does not have to be random. So if the females are more likely to mate with males with a certain allele, which might affect their phenotype, their courtship song, and Drosophila, etc., then one would expect that the population would change over time. The allele frequency of the male, which is more successful in mating, will increase over time. All right, so if it's 0.1 in this generation, in the future it might be 0.2, and then 0.4, and then 0.6, etc., simply because these males are more likely to foster progeny than other males. And so there are many ways in which phenotype is evaluated at the time of courtship. So for example, in birds have coloration patterns. Birds make songs. Male turkeys can spar with each other. Here's a frog. This is a male making a courtship song, etc. And so once again, if mating is not random, then we would expect that the allele frequencies change over time. Okay, so there are also then courtship behaviors we see in in birds, etc. I ask my students just to liven up the class. Like, so if you were to look at one of these males, or in the next video, one of these females, if you pretend that you're in this case, a female velociraptor, will you prefer one of these males? And then the same, true when I reverse it. Just the idea that if mating is not random, then we would expect that populations would then change over time. All right, populations would evolve. Evolution simply means change over time. Now, obviously, we can see this change at separate levels, and the next topic is speciation. So, there's more to this conversation. But very simply put, evolution is simply change over time. Natural selection can affect allele frequencies. So if you think of, I don't know, it could be the previous examples use pigmentation, but it could be, the size of the lungs or the gills. It could be the endurance in flight. It could be disease resistance. It could be muscle distribution. It could be metabolic rate. It could be size. These sometimes matter. So not all of these individuals may survive, and their ability to hold their breath underwater, their ability to catch fish, their ability to fly south in winter, the ability, once again, to hold their breath underwater and to catch fish, these matter. And so if not all individuals are going to survive, the environment may favor certain phenotypes. There may be a selection, a natural selection for more endurance, better vision, better disease resistance, a coloration which attracts a pollinator, which then allows you to make more seeds, etc. So natural selection then can affect populations. So if natural selection acts on, say, the dog color genes, then we would expect one to change. So for example, let's say that having one coloration in this case makes it easier for you to avoid predators. Well, obviously, if you're less likely to be eaten, you are more likely then to have offspring. Those which are more likely to be eaten by a predator will have fewer offspring. And so then allele frequencies would then be expected to change based on the natural selection due to predation. Now, just, pigmentation in, say, the dog color coat was one example, but this would obviously affect, your endurance in swimming, size that might affect your ability to swim into small crevices that would avoid the predators, etc. And so once again, in my class, I then say, well, what would we expect here? So once again, if there's 3 different colors in the velociraptor population, what would then happen if we get, predation? How would this affect the allele frequencies? Actually, let me get back to that. So first, let us look at this scenario where we have 3 different colors of velociraptor and they're running away from a predator. Now, notice the color of the background. And if this predator is trying to lock on one of them and follow that one, do you think any of these are more likely to blend in? Because if they blend in, here's hypothetically, that would be an easier way to avoid the predator. The predator would be more likely to choose the one that stands out. So in that example, was there one allele that was likely to now increase in frequency? Were there alleles which were likely to decrease in frequency? And the same thing here. All right, if a predator is more likely to prey on one which stands out and less likely to prey on one which blends in, once again, would any of those be then likely to change over time? So natural selection, like the natural selection from predation, this can cause allele frequencies to change over time. One would then become more common or less common. Now, something I'll just continue to bring up. If a population is infinitely large, if one dies from a predator, that really doesn't affect the population all that much. But some populations are small. Imagine we're on a small island and there's only 3 velociraptors. If predators prey on one, then that dramatically affects the population because it was so small. So events like this are going to have a greater effect in smaller populations. Even if they are random, even if this predator is colorblind and can't distinguish between these 3 colors, in a small population, if only one of them is blue and the predator eats the blue one, then that really does affect the allele frequencies in the next generation. The next generation's allele frequencies will change. There will be a change over time, especially in small populations. This is what's known as genetic drift, when random events, especially in small populations, change the frequency of alleles frequencies. Other things that I mentioned as well, mutations, always occur. But if mutations are not occurring equally at our two alleles, so let's say that, in the dog alleles, that the dark brown allele is more likely to mutate than the lighter allele. And there are examples of that, certain gene sequences can be viral insertion sites. They can be close to chromosomal breakpoints. Some nucleotides or some amino acid codons can more frequently change from one to another. If you've got these trinucleotides, that can more easily amplify. So I'm just saying that the DNA for one allele might be more likely to change than the DNA of a different allele. We know of examples for this. There are lots of causes of mutations, as you see here. So let's imagine that the rate of mutation is not all equal. If one of these alleles is more likely to mutate. All right, so for example here, the two blue parents, they had blue offspring. The two green parents, my fault, the two tan parents had a tan offspring. But notice that these two green parents, they had a blue offspring. If in this population, the mutation rate of green alleles becoming blue is higher than the reverse mutation rates of other alleles becoming green, then as we go forward in time, all right, then the frequency of green will go down, all right? So the allele frequency will change because of variations in mutation rate. And so, here is another example. Now, very often, members of your population, they could migrate out and go to a different, region. That could change allele frequencies through emigration, or individuals from another population could migrate in, and that would change allele frequencies. And so, in the United States, just to pick an example, if you could take allele frequencies for, say, hair color or blood type, well, there was a time when it was different tribes of Native Americans inhabiting the Americas, and allele frequencies would have been such and such. But then you had various groups of Europeans, and then tragically, they brought Africans enslaved as well. That would have changed. And then over time, there would be migrations from different parts of the world. So at this time, that a potato famine or war over here would have brought more migrants, from, this part of Europe or this part of Asia or this part of Africa or South America, etc. And so therefore, one could then ask, has the frequency of, hair color genes or blood type genes, has that remained 100% the same? No. As new groups join the population, allele frequencies change. Now, that doesn't have to be the case. If the group which is immigrating has all of the same alleles in the same frequency as the population which is already there. So immigration doesn't have to change allele frequencies. But it could, all right, because if the group coming in has, say, more green alleles, then the population going forward would have more green alleles than it once did. And so immigration and emigration can change allele frequencies, can introduce new alleles, as you could see here as well. So there are all sorts of scenarios, and natural selection can affect a lot of things. So you can have natural selection due to predation. You can have natural selection due to disease, etc. Now, this can affect any population, but a population which is infinitely large. If a few individuals get sick, then that has a minimal impact. But in small populations, that would have a much greater effect. So then we could consider two things. One, in a population, in a huge population, one which was infinitely large, it would matter if a new disease is coming about and say the green individuals are more likely to get sick. So, once again, we vary in our alleles. Some of our alleles make us more vulnerable to heart disease or asthma or flu, etc. So, alleles can certainly affect susceptibility to disease. So in a big population, a disease can change the frequency of alleles. Even if the green allele doesn't affect your susceptibility to a disease, in a small population, dumb luck can. So let's say that 3 individuals in this population of 6 die out of disease, and it has nothing to do with the color of their scales. But nevertheless, just because by dumb luck the disease happened to get both of the green ones, the population will then be different in the next generation. So one, any type of selection, such as selection because of disease, can cause alleles to change over time, if the alleles then affect disease resistance. But even if it doesn't, in small populations, just dumb luck, if you have a very small population and just by dumb luck one individual gets struck by lightning, then the allele frequency of the next generation is different, all right, simply because now one individual is no longer there to pass on their genes. Once again, that's called genetic drift, when random events can cause allele changes in a population. Now, let's say that a small population is devastated by a disease so that only 2 individuals live. All right, let's say these 2 individuals are now the parents of all subsequent generations. Well, that's what's called a genetic bottleneck. Bottlenecks don't have to go down to 2, but if you have a big population, something happens, an environmental change, a new disease or something, and the number of individuals then shrinks, then this small population, then they are the ancestors of all subsequent generations. That's a genetic bottleneck. And so genetic diversity would greatly decrease. And so one might notice that. So for example, I've read that, for example, with cheetahs, what I've read in scientific literature is that they don't have as much genetic diversity as other cats. All right, there aren't that many alleles and variations. Thus, it would seem that in the recent past, cheetahs underwent a genetic bottleneck where most of them died. All right, and then there was a small number of survivors, which are then the ancestors of the cheetahs, which we have today, for example. That same principle can then apply because individuals can migrate. So for example, here you can see a whole lot of fish at one reef. Well, maybe 1 or 2 of these decide it's too crowded, they swim somewhere else. They find a new reef where there's no grunts living, and they found a new population there. Well, let's say this one and this one end up at a new reef. Then 2 generations from now, all of the offspring at the new reef would be their offspring. So whatever their alleles are would then be found in the other reef. Sometimes populations are founded by small groups. So for example, it's hard to get around natural barriers like waterfalls and mountains and big rivers. So sometimes it's just a small number of one big group that make it across the mountain range, the big river, etc. And then the new population is then determined in large degree by the members who did make it across the river, etc. So this is what is known as the founder effect. So if we have, say, these dogs, look at the big population, look at all of the alleles. But if only a couple of them then make it across this river, then these are the ones who found the new population. The alleles of the next generations on this side of the river will all be determined by them. So they won't look like the original population. They will look like the founders of the new population. That's what's known as the founder effect. So if you go to a continent or an island and you say, oh, there's a tree, whether it be something like the frequency of, say, black hair or blood type O or some disease, this disease has a higher frequency on this island, or fewer people from this part of the world get this type of cancer than this part of the world, then that could be due to the founder effect. If the new population is founded not by a representative set from the original, but from only a couple of individuals, then the next generations will resemble the founders here. Before going into the different types of selection, just in my labs, I have some illustrations in an exercise which try to demonstrate some of this in fly populations. So if you remember, there was the individuals who were yellow and blue and the heterozygotes were green, that they were in between the two. Well, there are cases of that, say, in flies, where Drosophila melanogaster, the wild type condition, has a tan body. There's a mutant which has an ebony body, and the heterozygotes between them have some kind of a mottled appearance here. And so then if you were looking at P's and Q's, this individual would have two P alleles, this individual would have two Q alleles, and then this individual would have one of each. They would be P and Q. And then you could look then at a population and say, here's two P's and two P's, because they're both clear. Here we have two Q's and two Q's, but then here you have heterozygotes. And so once again, you could notice that the population isn't changing over time, or it is. You could calculate the number of Q's, the number of P's, etc. One other thing that you can do is you can say, let's say the population starts off with 50-50. So 50% of the population is P, 50% of the population is Q. And I apologize, because I teach genetics, the heterozygotes are actually a little intermediate, and I tried to be true to life. I shouldn't have. I just should have been more just more consistent artistically. So these are heterozygotes, but so is this one and this one. The ebony heterozygotes, there can be some variation. Not that you care. I tried to be true to life, but I have to explain that to my students. I need to make clearer illustrations. My bad. So let's imagine that the original population had P was 0.5 and Q was 0.5. But then populations then leave, all right? And so notice that this would affect this population because they've lost two P's in this individual and a P and a Q in this individual. So this population has lost P's and Q's. But then if these found a new population, notice that in the new population that there are 4 alleles, 3 of which are Q, and I'm sorry, 3 of which are P and 1 of which are Q. So both of these populations will see a change. So this group is losing 3 P's and a Q. But because it's a big population, the change will be smaller. But here, because they're founding a new population, the new population will then have 75% of the alleles will be P and 25% will be Q. So there will be a bigger change in the next population. And the same thing would occur here. What would be the change in this population? If one individual with 2 Q alleles and an individual with a P and a Q leave, well, that will affect not only this population, but then also the next population. But because small populations can be affected by small changes to a greater extent, we would see that in the founders of the new population. This would have a greater effect on the new population, which would now be 75% Q and 25% 25% P. And so just, I'll get back to the graphing at the very end. And so then this is something, that you could see, I use people, I use dogs, but one could see here as well. In the velociraptor, I asked, given the different backgrounds, would this affect predation to a differing degree? Well, the same thing here. If some fruit flies have a lighter body color and some have a darker body color, if they, a population was then adapting to come out at night, or if, they were present on fruit which had a light color, would this then affect them? You could apply the same things, here. Okay. The last major topic is when I use the term natural selection, we often distinguish between three different kinds of natural selection. One type of natural selection tends to select for the average phenotype in the population. So let's say that populations vary. Let's say they vary in size or they vary in color, and there's an average. Well, if those which have the average condition are the most likely to find food, the most likely to survive, then natural selection would favor the average condition. And so if the average is favored, then the odds are that the average would tend to stay the same over time. This is what's called stabilizing selection. So if natural selection makes it less likely for the very large dogs, say, to find food, or the very small dogs to find food, but the intermediate sized dogs, they have the best chance, then when you come back in the future, you would expect to find the median in the population, the average size, to be about the same. Because natural selection is working against the extremes, it would keep the average about the same. That's what's known as stabilizing selection. Okay. But natural selection can also then work in other ways as well. Let's say that the small dogs find it easy to find food, but the big dogs, they find it the hardest. So the big dogs are less likely to have offspring. The small dogs are the most likely to have offspring. This is then what's known as directional selection. One extreme will be selected against, and then the average of the population will shift. Over time, this population would be expected now to become smaller because more and more small dogs are having offspring, and fewer large dogs are having offspring. And the same could also apply in the opposite direction. So directional selection could go to either extreme. Here, the small dogs are having the most trouble finding food, and the large dogs finding it easiest. And so here, the population would tend to increase over time if the large dogs are having more offspring and the small dogs are having less offspring. We can also then have what's known as disruptive selection. Let's say in our big population, small dogs are finding food, large dogs are finding food, but intermediate-sized dogs are having more trouble. This would tend to encourage the formation of fewer dogs in the middle, and then more dogs which are either small or large. And this could then, over time, lead to two different groups. So from one species, from one population, we could then get two different populations adapted to different food sources, a small and a large group. In some of the activities for my classes, we imagine how selection might affect the beak width of finches, which then can correlate to the seeds that they can feed on. So the thicker your beak, the thicker the seeds that you would be able to crush. And so then I, have an activity where we look at finches, and we can actually measure the width of their bills with a little ruler. And then we imagine that there is a population where you have average seeds, thicker seeds, and softer seeds. And if it is the average seeds which are most common. And this type of finchbill, which is best adapted to feeding on these average seeds, then one would expect to see stabilizing selection, where the bills which are adapted to the average seeds would stay most common in the population. So if that's already the average, then the population would keep this average over time. That shape of the population, curve would then continue. So that's what's known as stabilizing selection. But then there could also then be examples where we see directional selection, where now the environment changes, and now the majority of the seeds are the thicker seeds. And it could be then this finch which is best able to feed on the thick seeds. This finch which is best able to defend nests or lay the most eggs, etc. And so over time, the finches adapted to softer seeds, they would become less and less common, where those adapted to the thicker seeds would become more and more common. The population would then change over time. So instead of the average condition being the most common, we would see the population shift in one direction, where more and more individuals were now getting beaks adapted for the thicker seeds. Or perhaps one could imagine the reverse scenario, where the population becomes more adapted to thinner seeds. Or if there's a mountain range which separates the population so that thick seeds are more common on this drier part of the island, while then thin seeds are more common on the wetter part of the island. That on two parts of the island, you could then actually have different kinds of selection. And so on one side of the island, thicker seeds are being favored. On the other side of the island, thinner seeds. But on then the island in general, we might actually then see a disruptive type of selection, so that we're actually getting two different populations on this island, and the average condition is becoming the least favored. So natural selection will favor those individuals adapted for certain environmental conditions, but then that can vary based on based on the environments. And so therefore, we could, then look at, various graphs, and say, what type of selection do I think is acting, here, if the extremes seem to be favored, or here, where it, this early one might be disruptive selection, where here it seems to be directional, where the lighter beans seem to be favored, etc. So certain graphs could be suggestive of different types of natural selection. One final point is that, in population genetics, there's certainly more that needs to be considered. One additional trait is that I was speaking of alleles as if they only affect one trait. So the dog color allele, that was the only trait, given, and affects pigmentation, or, the flies, or whatever. But we know of, other scenarios where traits can be affected by many genes. So for example, height is not just one gene, it's many genes acting together. So that would obviously complicate natural selection if the one trait is being acted on many genes in polygenic traits. Or in these examples here, there's something known as pleiotropy, where one gene can affect many traits. And so how does natural selection act? Well, once again, it's more complicated, because natural selection is acting on a gene which affects this trait, this trait, this trait. So while one might affect one aspect of physiology, then there would be other considerations as well. So this is just an introduction into some population genetics and into the application of the Hardy-Weinberg equilibrium.